Before we go on to EHP, let me just say that there's this thing between SPF and SPR, and Kona Bay has always focused on SPF. And beginning in 2011, we were interested in finding whether there was really a white spot resistant line from South America. And one of the reasons was that in GAA, we actually published a paper by a group from Panama where they did lab trials with Dr. Leitner showing that they did indeed have white spot resistance. And so we were curious, and so beginning in 2012, we took samples of shrimp from Mexico, Honduras, Panama, and Ecuador, and we did white spot challenge trials at Dr. Leitner's lab, and we proved to ourselves that indeed there was white spot resistance. And then in 2013, we went through a process of primary and secondary quarantine. It took about a year and a half going through all the proper channels, and we got one of those stocks into Hawaii. And at the same time, Mexico imported some shrimp from Ecuador, and they also imported those shrimp to get white spot resistance. But to everyone's surprise, those broodstock were also resistant to EMS. Just blind luck, really. In Ecuador, when white spot hit in 1998 or 1999, they had no way to implement biosecurity. So in Ecuador, they just began breeding the survivors of a white spot outbreak every generation, and now it's 16 years later, and their animals are very resistant to white spot. But actually, if you think about it, they're breeding for whatever diseases are present in the pond. So whatever bacterial diseases were present in those ponds, their shrimp also developed resistance to. So in a way, it's not so surprising that that stock is not only resistant to white spot, but to other diseases, including bacterial diseases. So at Kona Bay, we focus on the SPF approach, but we're very intrigued by the success of Ecuador with an SPR approach. So here in Malaysia, we're trying to work with Blue Archipelago and study the ability to develop tolerance to diseases by working right in the presence of the diseases. So we want to see if we can improve the EMS resistance and maybe also the EHP resistance by breeding right in the presence of the disease. So when you hear us talk about SPR, it doesn't mean that we don't believe in SPF. We want to try both approaches. And SPR has a few advantages over SPF in terms of the speed of progress that you can make. Because not only can you test family advantages, you can also utilize individual differences in performance, which you cannot do in an SPR approach, or in an SPF approach. Anyway, we can get more into that if you like. Now let's go to the EHP. So once again, I think Robin's already covered this very well. It's a spore-forming parasite. It impedes digestion, so it causes slow growth. Next slide, please. I found an interesting book about microsporidians, and there are a few diagrams in there, and this schematic diagram on the right, let's see, it's very interesting because it shows the thick wall as two layers, protein on the outside, chitin on the inside, and then that coiled thing that looks kind of like a rib cage is actually a long tubule And if you see the tubule here where it's extended, that's actually how this spore injects its germplasm into the host cell. In fact, some of the literature calls it a harpoon. It's like a long spear that's hollow. They call it it's like a combination of a harpoon and a hypodermic syringe. And so what actually happens is when the spore gets in the presence of the right environment, let's say in the gut of a shrimp, its vacuole, right there, this big gland in the shrimp begins to swell. And it's just like a CO2 cartridge. It builds up huge pressure within the spore until it finally ruptures, and that tubule everts out like a harpoon and penetrates the host cell and injects the genetic material. Next slide, please. So it works like this. You see the spores on the left, and then the tubule everted. The genetic material goes into the host cell. Then that genetic material propagates, multiplies, and then the spore walls form, and then the cell ruptures, and then it does it again and again and again until it begins to really seriously affect the digestion of the shrimp, because all of this is happening in the epithelial cells of the hepatopancreas. Next slide, please. So at the goal meeting in Ho Chi Minh City 2 years ago, Dr. Flegel from Mahidol University in Thailand presented this slide. And what he was explaining was a study in Thailand looking at 200 Thai ponds to see what is the incidence of EMS or APINs. As you can see on the left side, it was 24% of those 200 ponds had APINs. But the remarkable thing is he found every one of those numbers of a sample that's highlighted in yellow is one that also had EHP. And so you can see on the right there that EHP was actually double the incidence of EMS in Thailand in 2014. It was already much more prevalent than EMS in Thailand. So that's what's been happening behind the scenes. It's a very, call it insidious or kind of a secretive disease. Very hard to detect, and it's gone unnoticed. And once it establishes in a hatchery, everything that passes through that hatchery is infected, and one hatchery can stock many ponds, so it's easy to see how it multiplies. Do you want to say a few words about that?